I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimate to you. In the last session, we ended up with mechanism of action of eicosanoids. In today's session, we will start with organs on which prostaglandins act. Then we have leukotrienes and lipoxin. And lastly, we have synthesis of different phospholipids, cardiolipin, plasmalogens, and ceramide. Good morning, everyone. Let us begin the topic. Saturated fatty acids versus unsaturated fatty acids. Saturated fatty acids, there is an example like stearic acid, whereas unsaturated fatty acids, there is example like oleic acid. In saturated fatty acid, it is found in coconut and butter, whereas in unsaturated fatty acids, there is olive oil and sunflower oil. In saturated fatty acids, the hydrocarbon chains is in single bond and it is straight chain, whereas in unsaturated fatty acid, it is at least one double bond and it is bent chain. Saturated fatty acids have a high melting point, whereas unsaturated fatty acids have low melting point. In saturated melty acid, rancidity is low, whereas in unsaturated fatty acid, rancidity is high. In saturated fatty acid, there is increase in the LDL, whereas in unsaturated fatty acids, there is decrease in the LDL. In saturated fatty acid, there is 10% calorie intake in food, whereas in unsaturated fatty acids, there is 30% calorie intake in food. In saturated fatty acids, excessive consumption may cause atherosclerosis and heart attack, whereas in unsaturated fatty acid, it can reduce cholesterol levels. And clinical applications of prostaglandins. First, effects on CVS. Prostacyclin or PGI2 is synthesized by vascular endothelium. Major effect is vasodilation. It also decreases platelet aggregation and has a protective effect on vessel wall against deposition of platelets. But any injury to the vessel wall would decrease PGI2 synthesis and so that platelet aggregation occurs due to increased thrombus formation. Thromboxane A2 is the main PG produced by platelets. The major effects are vasoconstriction and platelet aggregation. Prostacyclin and thromboxane are in opposing effect. Prostaglandins decrease the blood pressure. In this chart, we can see that there is prostacyclin versus thromboxane. In prostacyclin, the structure is cyclopentane ring, whereas in thromboxane A2, the structure is oxygen ring. In prostacyclin, the site of formation is endothelium, whereas in thromboxane A2, the site of formation is platelets. In prostacyclin, the cyclic AMP level is increased, whereas in thromboxane A2, the cyclic AMP level is decreased. In prostacyclin, the platelet aggregation is inhibited, whereas in thromboxane A2, the platelet aggregation is enhanced. In prostacyclin, vasodilation is seen in blood vessel, whereas in thromboxane A2, constriction is seen in blood vessel. In prostacyclin, relaxation seen in bronchioles, whereas in thromboxane A2, constriction is seen in bronchioles. Effects on ovary and uterus. PGF2 increases the uterine vessels. Hence, PGF2 is used to be for medical termination of pregnancy. Yet another use is inducing labor and decreasing postpartum hemorrhage. PGs are involved in luteinizing hormone-induced ovulation. In cattle, if PG is given, the luteolysis takes place and animal goes into estrus. Coming to respiratory tract. PGF is a constrictor of the bronchial smooth muscle, whereas PGE is potent bronchodilator. PGE series are used in aerosol for relieving bronchospasm. Coming to effects on immunity and inflammation. PGE2 and D2 produce inflammation by increasing the capillary permeability. Erythema, erythema and wheal are produced at the site of injury. Moreover, PGE2 reduces both T and B cell functions. PGE2 is a sleep promoting substance. Next, coming to effects on gastrointestinal tract. PGs in general decrease the gastric secretion and increase intestinal motility. The inhibitory effects of gastric secretion are used therapeutically in treatment of acid peptic disease. But diarrhea may be an unwanted side effect. Coming to metabolic effects. The metabolic effects may be through the action of hormones by the modulating effect on cyclic AMP production. Prostaglandin E2 decreases lipolysis but increases the calcium mobilization from bone and glycogen synthesis. Coming to leukotrienes. They are produced from arachidonic acid. LTB4 is produced in neutrophils. It is the most potent chemotactic agent. The number 4 denotes that there are 4 double bonds in the structure. The 12 lipoxygenase in platelets produces 12 HETE, whereas 15 lipoxygenase in eosinophils produces 15 HETE. In this diagram, we can see arachidonic acid is converted into 5-hydroxy picosatetroinic acid with the help of enzyme 5-lipoxygenase. Then 5-HPTE is converted into leukotriene A4 with the help of an enzyme LTA synthase. Then this leukotriene A4 produces leukotriene 4 with the help of glutathione. Then this leukotriene A4 produces LX4A4 or lipoxin A4. It can also produce leukotriene B4, then leukotriene D4, and finally with the removal of glycine molecule, it forms leukotriene E4. Reacting substance of anaphylaxis contains leukotriene C4, D4 and E4. They cause smooth muscle constriction of bronchioles, increase capillary permeability, activate C leukocytes and produce vasoconstriction. SRS is the mediator of hypersensitivity reactions such as asthma. Coming to lipoxins, they are a group of compounds produced by leukocytes. They are conjugated tetraines. They are formed by sequential action of 5 and 15 lipoxygenase. Lipoxin A4 is the most common variety. It is anti-inflammatory and it decreases immune response. Coming to synthesis of glycerophosphatides. Phosphatidic acid is an important intermediate in the synthesis of phosphoglycerides as well as triacylglycerol. The phosphatidic acid itself may be produced from glycerol 3-phosphate or dihydroxyacetone phosphate. The synthesis of glycerophospholipids can occur either by activation into CDP choline, CDP ethanolamine, or by formation of active diacylglycerol and CDP diacylglycerol. Coming to synthesis of phosphatidyl inositol. In the CDP diacylglycerol pathway, phosphatidic acid plus CDP gives CDP diacylglycerol. The CDP diacylglycerol can react with the alcoholic group of serine or inositol to form the corresponding phosphoglyceride and releasing CMP. Phosphatidyl inositol undergoes further phosphorylation by a specific kinase. It thus forms phosphatidyl inositol diphosphate, which acts as a signal transducer. Synthesis of phosphatidylcholine. The major pathway for lecithin and sapharin synthesis, especially in liver and brain, involves the activation of choline and ethanolamine to phosphorylated derivative and then to form the CDP derivative. Finally, phosphocholine or phosphoethanolamine is transferred to diacylglycerol to form corresponding phospholipid. Phosphatidylserine can also be formed from phosphatidylethanolamine by a base transfer reaction. The addition of an arachidonic acid to the second carbon atom of glycerol may be achieved by removal of fatty acid already present in the action of phospholipase A2. Here in the diagram we can see how phosphatidylcholine is synthesized. First, choline plus ATP gives phosphocholine plus ADP in the presence of enzyme choline kinase. Then phosphocholine plus CTP gives CDP choline plus PPI in the presence of enzyme phosphocholine cytidyl transferase. Then 1,2-diacylglycerol plus CDP choline gives phosphatidylcholine plus CMP in the presence of ethanolamine phosphotransferase. In this diagram, we can see how phosphatidylethylamine is synthesized. Ethanolamine plus ATP gives phosphoethanolamine plus ADP in the presence of enzyme ethanolamine kinase. Then phosphoethanolamine plus CDP gives CDP plus ethanolamine plus PPI in the presence of phosphoethanolamine cytidyl transferase. Then 1,2-diacylglycerol plus CDP ethanolamine gives phosphatidylethanolamine plus CMP in the presence of enzyme phosphotransferase. Then phosphatidylethanolamine plus serine gives phosphatidylserine plus ethanolamine. This is followed by an addition of an arachidonic acid molecule from arachidonyl-CoA by a specific acyl transferase, or from another phospholipid molecule by LLAT, that is lysolecithin acyl transferase. For the formation of pulmonary surfactant, dipalmitoyl lecithin and palmitic acid is added by the same reaction. Lysophospholipids are often the source of fatty acids for such purposes. In this diagram, we can see cardiolipin and plasmalogens. Cardiolipin, it is formed by reaction between CDP diacylglycerol and glycerol 3-phosphate. Plasmalogens, they are formed from dihydroxyacetone phosphate by acylation and addition of choline and ethanolamine to form a CDP derivative. The final step in the reaction offer desaturates on the alkyl residue in the first carbon atom, giving molecular oxygen and NADPH to give a plasmalogen, phosphatidylcholine and phosphatidylethanolamine. Yet another important enzyme acting on lecithin is LACT, which transfers a PUFA from second carbon of glycerol to cholesterol, forming lysolecithin and cholesterol ester. Coming to synthesis of sphingolipids. Sphingolipids may be of two types: phosphosphingolipids and glycosphingolipids. Both types are important components of biomembranes as well as brain. Most important sphingolipid is sphingomyelin. The glycosphingolipids may be cerebrosides, ceramide, oligosaccharides, and gangliosides. Coming to synthesis of ceramide. Ceramide is a basic structural unit of all sphingolipids. It is formed from a sphingosine and fatty acyl-CoA. Sphingosine is formed in endoplasmic reticulum from palmitoyl-CoA and serine in the presence of pyridoxal phosphate. We can see synthesis of ceramide. Palmitoyl-CoA gives 3-keto-dihydrosphingosine in the presence of pyridoxal phosphate. Then this 3-keto-dihydrosphingosine gets converted into dihydrosphingosine in the presence of enzyme reductase. Then dihydrosphingosine plus fatty acyl-CoA gives dihydroceramide plus CoA in the presence of enzyme acyl transferase. Then dihydroceramide gets converted to ceramide in the presence of enzyme dehydrogenase. Here ends our session. Kindly like, share and subscribe the channel. Thank you.